Hello. Today I'm going to go over cell theory lecture. And this is just an introduction to this history of identifying and categorizing cells. Okay, so let's just get started. We've got an image here, a microscope image of skin cells, different layers of skin cells. And you can see that the cells of different layers look different, but their basic structure is the same. All living things are composed of cells. Before the 1600s, people had many different ideas about what constituted the basis of life. So this idea right here, constituting the basis of life, it's giving us kind of a foundation of like what are living things made of. However, with the invention of the microscope, scientists could finally see that cells are the smallest unit of living matter that can carry out all processes required of life. So what we're saying here is nothing that's smaller than a cell or things that make up a cell would be considered life. Life is the smallest unit of a living thing. In this image here, we have a very early microscope, or a way at least of enhancing an image, looking at things closely. Most cells are too small to be seen without a microscope. The invention of the compound microscope in the late 1500s was a crucial step in the discovery of cells. The Dutch eyeglass maker Zacharias Janssen is credited with this invention, which used two or more lenses to magnify images to a greater degree than a single lens could. So in this, you'd have two lenses, I'm assuming one at each end. And when you look through the eyepiece here, you're looking down at whatever sample you have down here, and it enhances it. Since this was an early one, the detail was obviously not really great, so we have to improve on that. All right, so continuing on. In 1665, Robert Hooke used a 3-lens compound microscope to examine cork. So this is cork like you would find in wine bottles. Cork comes from a tree. So he put thin slices of that under a microscope. He observed tiny hollow compartments, which he named cells after small rooms in a monastery. That's what this image is showing, is thin layers of cork, and it's made up of these tiny cells, which she saw as like these little pockets, right? But couldn't see inside or what was happening with them. Hooke was only able to see the cell walls and empty space because the plant cells he observed were dead. So in this case, they've been long dead, and it was just the outside structure that he could see. Around the same time, Antoine van Leeuwenhoek developed a more powerful single-lens microscope. In 1674, he became one of the first to describe living cells when he observed single-celled organisms in pond water, which he called animalcules. So this is a normal thing to do in science labs, like elementary through high school, really, is you look at a drop of pond water because it has lots of microorganisms inside. And with any kind of basic microscope, you can see things swimming around in the pond water that you could not see with your naked eye. And we've got some examples here, but it was important that he was able to see living things. Things moving through the water and swimming through the water that were single-celled in nature. The continued improvement of microscopes over time allowed scientists to observe a wider variety of cell shapes and even see cells dividing, prompting new questions about the nature and origins of cells. So when they saw cells dividing, it's kind of like, well, wait, are cells constructed or are they replicated through division? Every time you make new discoveries, it seems like there are more questions that get presented after that discovery. All right. In 1838, Matthias Schleiden proposed that plants are made of cells. So we know that they had seen cells in cork, and we had seen individual things living in pond water, but now it's all kind of coming together to form this general idea. The following year, his colleague Theodor Schwann came to the same conclusion about animals and published the first statement of cell theory, which concluded that all living things are made of cells and cell products. So that's one important part of this cell theory. All living things are made of cells and cell products. This early theory, however, had to be refined because Schwann mistakenly believed that cells could form spontaneously. So forming spontaneously just meaning forming out of random stuff in an environment, in water, in the soil or whatever. And that there was no point of origin for the cells. So we'll find out that that's a little different. So cell theory initial, now let's get a little more specific. In 1855, Rudolf Virchow corrected Schwann's mistake, asserting that all cells come from pre-existing cells. This accumulated research led to the modern cell theory, one of the fundamental concepts of biology. So he said that all cells come from other cells, so that there's always this replication, not just made from scratch. Its major principles are, and here's the three major principles of the cell theory. Its major principles are: all organisms are composed of cells, which we see here. All existing cells are produced by other living cells. We see a parent cell dividing into two daughter cells, so increasing the number of cells through division. And the cell is the most basic unit of life. So we go from the most basic cells and then more complex forms of life: tissue, organ system, and organisms. All right, and then finally we start to distinguish between two main types of cells, the more detail we get about what cells are. Organisms are composed of two main types of cells: prokaryotic cells and eukaryotic cells. Both cell types have cytoplasm, a jelly-like substance that contains the building blocks of life. So that's all this jelly inside this blue stuff here. And both are enclosed by a membrane. We see cell membrane around both. However, the key difference lies in their internal structures. So there'll be some differences inside. Let's go over that. Prokaryotic cells do not have a nucleus or other membrane-bound organelles. So, prokaryotes, these are your basic cells. These are like bacteria. The DNA is floating inside of the cytoplasm just throughout. In eukaryotic cells, everything else that we think of pretty much, insects, people, dogs, fish, whatever, the nucleus is containing the DNA, and it holds the DNA. In contrast, eukaryotic cells have a nucleus that encloses their genetic information, as well as various other membrane-bound organelles that perform specialized functions. So that's for later, right? That's the next step in learning about cells. Eukaryotes can be multicellular or single-cellular, where prokaryotes are single-cellular organisms. So eukaryotes can be just the living thing, and that's just one cell, or more commonly multicellular, where lots of eukaryotic cells come together to form like people. Okay, I hope this gets you started. If you have any questions, let me know. Thank you very much.